Hello friends, again welcome to my channel. Today we are discussing about a process of phagocytosis. Preferentially within that we are dealing with which are the basic steps that are involved within the phagocytosis. Now, before starting with the phagocytosis process, it is important to know what is meant by this phagocytosis. Now, phagocytosis, it is a process for the engulfment of foreign particle and there is the intracellular killing of that ingested material. Basically, it is a Greek word, the meaning of which it is the to eat. Means the cells, these are responsible to eat this ingested material or to digest that ingested material. Within which the specific type of the cells, with the help of their plasma membrane, they engulf the larger particles, giving rise to an internal compartment which is called as the phagosome. It is also the one type of the endocytosis process, and preferentially those of the cells which are carry out the phagocytosis, these are called as the phagocytes. The main type of the phagocytic cells within our immune system includes the macrophages and the neutrophil cells. Now, the first initial step involved within the phagocytosis, it includes the activation of phagocytic cells and the chemotaxis. Now, majorly the chemotaxis, it is a movement of the phagocytic cells towards the site of injury, and preferentially this movement it is carried out due to the secretion of certain chemicals. These chemical substances, these are responsible for attracting these phagocytic cells towards the site of injury. These phagocytes, these are attracted and move toward the variety of substances, that is the chemical substances which are secreted from the site of injury, and this process it is called as the chemotaxis or the chemotactic movement of these phagocytic cells. Now, the resting phagocytes, these are get activated by the inflammatory mediators. These inflammatory mediators, these are including specifically the different types of the bacterial products, including the lipopolysaccharides, then the different cytokines which are secreted by the immune cells, then the prostaglandins and the complement protein. All of these factors, these are acts as an inflammatory mediators, and these are responsible for the activation of the phagocytic cells. This activation step, it is responsible to increase the metabolic and the microbicidal activity of the phagocytic cells. Now, once there is the activation of the phagocytic cells, these phagocytic cells, these express the more amount of the glycoprotein receptor on their surfaces, which help them to reach the site of infection, and majorly these receptors, these are helpful for the binding with the foreign particles, that is including the microorganisms. The neutrophilic cells, these are the first cells which are appeared at the site of injury, and then later on these are get replaced by the macrophage cells. Now, here within this figure we can observe the complete process of the phagocytosis. Here in figure we can observe the phagocytic cells. The bacterial cell, it is in contact with the phagocytes. Majorly the receptors, these are present on the surface of these phagocytic cells. There is the binding of this with this phagosome, then these are transferred within the phagosome, and then there is the intracellular killing of that bacterial cell, and then the soluble debris, converted soluble debris, it is get released outside the cell through the process of exocytosis. So, one by one we are describing about all of these steps. Again, here there is the descriptive explanation of the process of the phagocytosis within this diagram. Now, here again it is an early phagosome, phagolysosome, lysosome, these are present. Means whenever there is the intracellular killing of ingested material within the phagocytic cells, majorly firstly these are transferred within the phagosome, then it is transferred to the phagolysosome, and within that there is the complete degradation of the ingested material. Now, the second step involved within the phagocytosis, it includes the recognition of invading microbes. Now, it is the second step within the phagocytosis, that means the adherence of the antigen or the bacterial cell to the cell membrane of the phagocytic cells. This adherence it is responsible to induce or there is the induction of membrane protrusions within the phagocytic cells, that is the pseudopodia of the phagocytic cells, and these pseudopodia these are extend around the attached material and ingest them. Now these phagocytic cells again these are containing the various receptors which are responsible to help them to attach with the bacteria or viruses, and these are the example of these are including the pattern recognition receptor, which are in short called as the PRR. These pattern recognition receptors these are responsible for recognition of pathogen associated molecular patterns, that is PMPs. PMPs, that is these include the bacterial molecules, peptidoglycan, teichoic acids, which are the components preferentially present within the gram positive bacteria. Then the lipopolysaccharide mannans, flagellin, pillin, these are the components of the gram negative bacteria, and also the bacterial DNA which are present within the every bacterial cell. Now the second type of the receptor it is the Fc receptor. Now these Fc receptors these are present on the surface of the macrophage cells and the neutrophil cells, and preferentially these are having the binding site for the Fc receptors of the immunoglobulin molecules. Whenever there is the entry of pathogens inside our body, the immunoglobulin molecules these are attached with that particles, and majorly these attached immunoglobulin molecules these are responsible to increase the phagocytosis or the rate of phagocytosis of this material, because majorly these are responsible to increase the size of these particles. Due to the attachment of these immunoglobulin molecules there is the increase within their size, and due to that this particulate material it is easily get engulfed by the phagocytic cells. Means here there is the enhancement of the phagocytosis, and this process it is called as the opsonization, and here these immunoglobulin molecules these are acts as an opsonins. Here within this figure we can observe these pattern recognition receptors which are present on the surface of the phagocytic cells. This yellow region it is considered to be the, here these are the surface of the phagocytic cells, and the red colored bacterial cell it is get attached with that of the pattern recognition receptor present on the surface of the phagocytic cells. Here second one it is the, here there is the receptor for the IgG, that is the immunoglobulin molecule. For this Fc region of the IgG antibodies which are attached to the bacterial cell, the receptors these are present on the surface of the phagocytic cells. Means here it is the process of opsonization. Now the third stage within the phagocytosis it includes the ingestion and the formation of phagosomes. Now once there is the attachment or adherence of the foreign particle or bacterial cell with the phagocytic cells the surface receptors, there is the polymerization and then the depolymerization of the actin filaments, and which send the pseudopores out to engulf the microbe. Means due to this polymerization and the depolymerization of the actin filaments these are responsible for changing the shape of the pseudopodia, and by means of which the phagocytic cells they are responsible to engulf the microbial cell or the any foreign particle inside the cell, that is inside within the endocytic vesicle which is called as the phagosome. Now this phagosome it then enters the endocytic processing pathway. Now the fourth step within the phagocytosis it includes the formation of phagolysosome. Now again within this there is the early phagosome. Within the phagosome there is the pH it is up to the 6 to 6.5. Again when it is get transferred to the interior of the cell there is the reduction within the pH of this, and then within this pathway this phagosome it moves towards the cell interior, and within that it interacts or it fuses with the lysosome, and then there is the formation of phagolysosome. As a result of fusion between the phagosome and the lysosome there is the formation of phagolysosome. Now the next step it is the microbial killing and the formation of residual bodies. Now the lysosome which is fuses with the phagosome, this lysosome it contains the major digestive enzyme, that is the lysozyme, and a variety of different antimicrobial and cytotoxic substances, along with that different hydrolytic enzymes which are responsible to destroy the phagocytosed microorganisms and the microbial cells. These microorganisms, these are killed either by the oxygen dependent or by the oxygen independent mechanism. Means the intracellular killing of the ingested material within the phagocytic cells, it is carried out by using any of the one pathway, that is either there is the oxygen dependent killing or the oxygen independent killing of that ingested material. Now the last step within the phagocytosis, it is the elimination or the exocytosis. Means after the intracellular killing either by the oxygen dependent or oxygen independent pathway, the digested contents of the phagolysosomes, these are eliminated from the phagocytic cells through the process which is called as the exocytosis. Means these are the different steps which are involved within the phagocytosis. Now the details, that is how there is the oxygen dependent pathway, what are the chemical substances or what is the mechanism for oxygen dependent killing of the ingested material, then the oxygen independent killing of the ingested material, that part we are discussed within my next lecture. If you like this video, please subscribe my channel and also share this video with your friends. Thank you.